Hello, everybody. This is Sheila Heymans speaking. Well, I want to welcome you to the European Marine Board's Third Thursday Science webinar series. As I said, my name is Sheila Heymans, and I am your moderator for today. I want to just go through a few quick housekeeping rules for those of you who have not been here before. When you come in, please make sure that you change your name so that it's clearly entered, so we know who's asking any questions. When we have questions at the end, you can ask your questions by using the Q&A, which you should see at the bottom of your screen. And when you do, please say who the question is for, and also what your organization and your country is, so that we know where the question is coming from. If you have any problems, please use any technical problems, please use the chat function. And please be aware that this webinar is being recorded and it will be available on the Marine Board website and on our YouTube channel. It is also being live streamed, I believe. So today our webinar is on microalgae, which is working for a cleaner planet. It's about waste valorization, resource recovery, and the circular innovation. It is related to a project that the European Marine Board is part of called Remediation, a lighthouse project under the Ocean Decade, under the Mission Ocean. And it is related to the chapter that we have in Navigating the Future 6 on ocean and fresh water. We have two speakers today. The first one is Luísa Barreira. She is an associate professor at the Chemistry and Pharmacy Department of the Faculty of Sciences and Technology of the University of Algarve. And she's also a senior researcher at CICIMAR in Portugal and co-lead the Marine Biotechnology Group. And then our second speaker will be David Liberti, who is a postdoctoral researcher in the remediation project at CICIMAR, also from the University of the Algarve in Portugal. So I'm going to stop sharing my screen, and I believe David will share the screen and Luísa will go first. So David. Here we go. Just waiting for full screen mode. Perfect. Thank you.
Thank you very much, Sheila, for the introduction. Thank you very much for attending this webinar. We will be discussing some part of our research done at the Center of Marine Sciences based at the University of Algarve in Portugal. Next slide, please, Davide. Okay, so just a few more details apart from what Sheila already has said. My degree is in chemistry. My background is applied chemistry, and then I did my PhD in environmental sciences and technologies, which you will see by the results that we are presenting. It's a background that comes very much in handy when you are working in wastewater treatment. I would also like to say that I'm one of the found members of the Green Collab, which is a non-profit collaborative platform that aims to bring together the academia and the industry. And just to say that I've been working with microalgae for the past 16 years. Davide, maybe you can present yourself.
Sure.
On the next slide.
Yeah, hello everybody. I'm Davide Liberti, and as said before from Sheila, I'm a postdoc at CICIMAR, coordinator of the scientific area of bioremediation and wastewater treatment at Mar Biotech. And I did my studies in the University of Napoli Federico II, where I carried out my PhD in biotechnology and my researcher in microalgae for the past 6 years.
Okay, so just a brief presentation of the Mar Biotech group. We work with marine organisms, microalgae, some seaweeds as well, and also with the halophyte plants. But we work in several aspects of the microalgae, so not only the bioremediation and wastewater treatment that we will be presenting here today, but also on the development of new strains, from the isolation to the culture scale-up and to the improvement of those strains, and also on the applications ranging from biofuels to biomedical applications as cosmeceuticals, pharmaceuticals, nutraceuticals, so several aspects of microalgal biotechnology. Next. Okay, so big introduction for what are microalgae, and here we include also cyanobacteria because these are microorganisms that can do photosynthesis. As you can see in the pictures, and these pictures were taken of microalgae isolated in the Algarve coast, and you can see the huge difference in shapes, in colors. You can see that some of them are truly unicellular, others are arranged in colonial organisms. The ranges in sizes in microalgae is huge. You can go from as small as 0.2 micrometers to 200 micrometers. And they produce a lot of interesting compounds, and some of them give them, for example, the different colors, which are because of the different carotenoid pigments that they produce. You can also see that some are more round, others are these long shapes, and they can group in groups of 2 or 4, or then these strands, which are very characteristic of the filamentous macroalgae. Next slide. In terms of cultivation, we can have 2 types of cultivation, the open systems and also closed systems. They have different uses, let's say. So open systems are usually cheaper, and we use it to produce bulk amounts of low commodities, let's say. So biomass with less value, because as they are open systems, you have less control over the contaminations, and so the product is usually with less quality. When you want to produce a niche organism that cannot outgrow competitors, for example, or when you want to produce something that is designed for food consumption, usually we choose closed systems where you have less risk of contamination, higher control over the quality of the biomass produced, but of course you have a very high initial costs. So the capex is very high on these systems, and you have high energy consumption as well. Next step, next slide. In terms of biodiversity, microalgae are one of the highest biodiverse groups of organisms. It is estimated, because we don't know all of them, but it is estimated that you can have as high as 800,000 species. Less than 50,000 are described, and when you move to industrial cultivation, less than 50 are cultivated at industrial scale. So there is a lot to do with microalgae. Next slide. In terms of composition, and the uses of microalgae are very much associated with their composition. Just doing the proximal composition in terms of carbohydrates, lipids, and proteins, you can see that you have a huge diversity. So there are some species that have very high amounts of lipids, and usually the profile of these lipids are very unsaturated, which makes this species very very good for food or feed. But then you also have some with very high amounts of protein, which you can suggest that maybe these strains can be used as a novel protein source. Then we have also the carbohydrates, and they can be used also to produce biofuels. So depending on the composition, there is a lot that we can do with these microalgae. Next slide. So in terms of applications, and as I was saying, depending on the composition, you can use these microalgae to produce the biofuels. You can also produce bioplastics. You can use them to food, medicine, also as to produce biofertilizers, biostimulants. But what we are going to talk today is about their application in wastewater treatment. So to produce microalgae, we need to give them sunlight, we need to give them water, and we need to give them food. And microalgal food, we're talking about nutrients. Nutrients as nitrogen and phosphorus, which are very abundant in wastewaters. So we can use the wastewater to cultivate microalgae. You do the treatment of the wastewater, and at the same time, you produce a biomass that has value for valorization, which makes microalgal treatment of wastewater perfect for a circular economy concept. Next slide. Okay, for the biofuels, what we need to do is then a cheap cultivation, because we never want to pay that much for a biofuel. And if you want to compete with the price of petroleum fuels, then you need to do it cheap. So cultivation will always be in an open system, and then we need to do the lipids extraction and the biofuels. And this is just an example of one of the biofuels that can be produced with microalgae. Next slide. For the bioremediation, what we are trying to change is the way that wastewater treatment is done. What you have here on the right side and also on the left side is a picture of what is the conventional wastewater treatment. And usually it contains a lot of steps. Most often it stops here on the secondary treatment, where you have already a very high reduction on the level of organic matter in the wastewater and also of nutrients. Sometimes you still have a tertiary treatment that can aid the final treatment, removing additional nutrients and also doing the disinfection of the water. So most of the studies that exist with microalgae are done with microalgae doing the tertiary treatment, removing more nutrients that were not yet removed in the secondary treatment. But there are already some studies that are replacing the secondary and the tertiary treatment using microalgae. Next slide. So in terms of legislation, we need to look at the European legislation and wastewater treatment. There has been some very recent developments, and these legislation for wastewater treatment in Europe came in November in 2024, late November 2024, so it's quite recent. And now member states have some time to put this in practice. What changes from the last is that there is an increased awareness and concern about the release of emergent pollutants. Wastewaters are one of the biggest routes of entry in our oceans and in the Mediterranean as well, of emerging pollutants. We are talking about polycyclic aromatic hydrocarbons. We are talking about polychlorinated biphenyls, pesticides, pharmaceuticals, and also the forever chemicals, the PFAs. So there are a lot of chemicals that go into our coastal waters because of wastewaters. Often that secondary treatment or the design that was in the last slide are not completely effective in removing those pollutants. And so it is not just the concern about removing the nutrients. Now we also have the concern of trying to prevent the emerging pollutants to go into the waters of being released. So we need to capture them in the wastewater treatment and prevent their release into the environment. What we are doing here is replacing then these conventional wastewater treatments with microalgae. Next slide. So our approach is to use the use of microalgae to do not only the removal of nitrogen and phosphorus, but also to bioaccumulate or sometimes do biotransformation of all the other pollutants that I was mentioning. In this biotransformation, it means that the amount, with both, the amount of these emergent pollutants in the released water will decrease. With the biotransformation, what we aim is that the results of the biotransformation are of compounds with less toxicity than the parent compounds. Next slide. Okay, these are some of the chemicals that can be removed by microalgae. I know it's very small. It's not our objective that you can look at all the compounds and their removal rates. What you need to look is the colors, red and yellow. And what they mean is that the studies that have been done with the application of microalgae to remove these contaminants from water has been extensively studied, but the largest majority of the studies done are being done at lab scale, not at pilot scale. And if you see those yellows are the few studies that were conducted at pilot scale outdoor, where we have all the light fluctuations from night to day and from the different seasons. We have the temperature drop at night. So we have a wastewater that changes composition daily and hourly as well. So the conditions that can be tested in the lab are good to see if really microalgae can do the removal, but they can never mimic the conditions that the microalgae will have when you go to the large scale. So that means that we need to go into the field and we need to prove that this technology can work. And that's what we have been trying to do for some time now. This is the first project that we went from the lab to the outside. The Green Tree project was conducted in the Algarve, and we used a different model of photobioreactors than the ones that I have just showed. Why? Because if you look at the small picture on the right, what you can see is those raceways. The depths of the water in those systems is around 20 to 30 centimeters. And it's that shallow because you need the light to penetrate in order to promote the photosynthesis. The problem is that they take a lot of space. And if you are going to do this to treat wastewater from a large municipality, you usually do not have enough area to devote to this. And actually, we occupy 10 to 20 times more space than a conventional wastewater treatment. And this has been one of the main reasons why you don't see microalgae still at work in doing wastewater treatment. The photobioreactors that we used in this Green Tree project were developed by a Portuguese biotechnology company, Blue Matter, and they have 3 times the volume per area ratio of the conventional microalgal systems. And so it makes them more applicable to do wastewater treatment. And I'm going to show you some of the results that we got with this project. Next slide. Okay, so what you can see here is the natural blooms development of microalgae. Natural blooms means that we did not inoculate the reactors with a specific strain. The strategy was a bit different. So we have the water with nutrients, and in this case, the water that was feeding these reactors was coming out of the secondary treatment, but it still contains 4 times more ammonia that should be in the water in order to be discharged into the environment. So that means that we still had a lot of nitrogen to feed these microalgae. And if you just let the water stay with light for a few days, what you get is on the picture on the bottom. And what you have is a mixed culture of microalgae. Of course, some bacteria, not a lot in this system because the organic matter had mostly been removed in the secondary treatment. So the chemical oxygen demand of this water was already very low, below 30 milligrams per liter, which is almost nothing. And so what we had was a microorganism consortium largely dominated by microalgae. So we did a survey. Of course, everything here, the water never stopped in the system. So it was continuous cultivation, continuous operational mode. And what we tested were different hydraulic retention times. We tested 24 hours, 48 hours. We also tried 12 hours. So that you can have like a notion what this actually means. It's like the average time that the water passes in the system. In a conventional bacterial system to treat wastewater, usually they use hydraulic retention times between 6 and 18 hours. So ours needs to be a bit higher because microalgae do not grow as fast as bacteria. And that is the main reason why you cannot use such low hydraulic retention times. But then that is also one of the reasons why you occupy so much space, because you need larger volumes, because the water needs to be there for higher, more time. Next. Okay, so what you see is on the right is the results that we get. I know it's very small, but you can see some gray lines, which are the legislation values. And you can see that most of the time it actually worked. So even using 24 hours of hydraulic retention time, we were able to treat the water and remove the nutrients enough to discharge. Where it failed was when we tried to use the 12-hour hydraulic retention time. And this is why this happened, because we didn't have a recirculation system of biomass, and 12 hours was not enough for the microalgae to replicate, to duplicate, and replace the biomass that was being removed from the system in the continuous cultivation. So that's why in the next projects, we used recirculation of biomass so that we can decrease the hydraulic retention time, but still keep the biomass in the system and do not have what happened here, which was a washout of the biomass. Next. So we also tried to see if there was an additional removal of the pharmaceuticals. And let's say that for some compounds, the conventional wastewater treatments with activated sludge or oxidation ponds, so bacterial treatments, they can remove for some compounds large amounts. The problem is that some of the compounds exist in the water at so much, they are so concentrated in the wastewater that even with good removals, the amount that comes out can still be toxic to aquatic life. So in this case, we could see that the water that was coming out of the secondary treatment still contained a lot of pharmaceuticals, but in many cases, we were able to reach 100% removal with the microalgae in this system. Next. Of course, then we are also concerned about the environment. And as good these systems can be because they just use sunlight, so they do not consume a lot of electricity, as natural processes they still have an environmental footprint. And what we wanted with this study was to compare the environmental footprint of these microalgae treatments with the tertiary treatment that this plant was using, that was a denitrification step done with bacteria. Now, to do nitrification with bacteria, you consume a lot of air because they need a lot of oxygen. Let me tell you that in this plant, 60% of all the electricity consumption was in that small system. Is that tank that you see behind our system? It's a very small occupied space, but it consumes a lot of energy. And in the bars that you see on the right, you can compare and see that the blue bars, which represent our green dune system, compared with the red bars, which represent the operation done at the wastewater, we were in all cases better. So our environmental footprint was always less. And the small reduction, it looks small reduction, but that reduction represents almost 70 to 80% less of environmental footprint than the conventional system. Next slide. Now, in order to get the circular economy, you need to do something with the biomass. So with the biomass that we produced, we did some only lab scale studies trying to see if this biomass could be used to produce biogas. So in this case, we tested the biomass produced in different months of the study, of the pilot study. And we could see that the yield of biogas production was not the same. So the biomass had different compositions, and that affected the production of biogas. We could also see that the biogas production was not as high as we were expecting. And in the graph in the right, you can see a higher production done by a pure culture, not pure culture, let's see, monoalgal culture that we were comparing with ours. So ours were a component, so a natural bloom. It was a complex consortium. And when we compared with the monoalgal culture, our system performed less. But still, biogas production is possible. Next. Of course, we were not controlling the composition of the culture. We were interested in seeing what were the species that were dominating. And what we could see was that the diversity was largely affected by season. That means that in summer we have higher temperatures, in winter we have lower temperatures, and that affects the composition of the consortium. And we could see that the diversity, so in terms of species richness, we have much richness in much diversity in spring, and then very, very low diversity in summer. But when we look at the evenness of this diversity, so when we take into account not only the number of different species, but also the abundance of those species in the consortium, then spring had very richness. It was very rich in diversity, but they were with a low evenness. That means that they were dominated by a few species. And those species that dominated in spring were not really microalgae. They were dominated by rotifers that were feeding on microalgae. And actually, often when we went to the pilot and checked things, we could see that the culture had collapsed because the rotifers had eaten all the microalgae. But after a few days without doing anything, and when I say a few days, sometimes it was only one day, when we would go back without doing anything, the consortium had recovered. And this is one of the biggest advantages of not using monoalgal cultures, but using the consortia, is that their ability to recover from these events. What we could see also is that, sorry, Davide, is that some of the consortia were more efficient than others in removing the nutrients. And we could see that, for example, the winter species were not as efficient as the summer species in removing the ammonium, mostly the ammonia. Now you can go on.
Sorry. Sorry for
Okay.
Okay.
So
I
then
will
we came
take
to the remediation project, and now Davide will explain what we are doing on this project.
Okay, so basically, yes, I'm gonna give you some insights about the remediation project. As said before, remediation stands for Responsive Hub for Long-Term Governance to Distress the Mediterranean Sea from Chemical Pollution. In this project, we are aiming to install three different bioreactors, pilot system, in three different places, which are Greece, Italy, and Turkey. And as you can see, there are so many different partners involved. And the aim is to tackle the emerging pollutants that are present in the wastewater even after the conventional treatments. The emerging pollutants, as Professor Luísa said, are defined as the compounds that are not currently covered by existing water quality regulations. As you can see, this definition is quite outdated because now the water quality regulations are taking into account those pollutants. So, but still, the conventional methods, treatments are not updated yet. The first demo site is located in Greece, in Elefsina, which is an enclosed bay, and the system has been installed in the Triaso wastewater treatment plant. In here, the inlet water for our system will be the water sourced after the secondary treatment of the wastewater plant. So basically, our pilot system will replace the tertiary treatment that is already present in the system. Then we have the Turkish demo site. The system has been installed 2 weeks ago. And in here in the Izmit Bay, differently from the previous demo site, here the wastewater is mostly urban wastewater. Meanwhile, for the Greek demo site, we had mostly industrial wastewater. Another difference in this demo site compared to the previous one is that the wastewater that we will use to feed our bioreactors will be wastewater sourced after only the primary treatment. So only after the main grid to remove the larger debris. Third demo site, also different in Italy, in Taranto. We will very different from the others because the system will not be implemented in an already existent wastewater treatment plant, but will be placed basically next to a nursery, and the source water will be the Galeso River, which is not so polluted river, but could accumulate some heavy metals in particular, especially from the farms and from the fields that are surrounding the rivers. This table shows basically which are the pollutants, the emerging pollutant that have been taken into account by the latest update of the legislation. As you can see, the list is long now with the latest update, but in the past it wasn't that long. These are some pictures of the installation of the Greek demo site. The system seems to be very difficult from some pictures. Of course, if you take into account all the pipes and the automation that is needed to make it run autonomously. But actually, the bioreactors itself are very easy to build because they are plug and play. There are just few pipes in between, and you are good to go, I would say. In the end, at the end of the installation, the system was built by 30 units with a total volume of 15 cubic meter. The consortium of the microalgae instead of the bioreactors took more or less 10 days to develop. The hydraulic retention time is set at 24 hours in order to respect what Luísa said before. And there are also 6 sensors that are used to make the system run automatically, with each 6 probes for pH, redox potential, ammonium concentration, turbidity, conductivity, and chlorophyll content. These parameters that are analyzed are also important not only to verify if the microalgae, the consortium, is actually working to remove the pollutants, but also to determine the quality of the consortium, because if the consortium could crash inside the system due to heavy rain or some environmental and weather effects that can happen, the system will recover, as said, but we need to monitor the level of nitrogen in order to adjust the flow of the system. And as you can clearly see from this picture, the picture on the left was the starting point of the system. In here, the water seems already quite greenish, but actually it's just an effect of the light of the picture, because the water was yellowish. There were no microalgae inoculated, but just after a few days and then in 1 month, the microalgal bacteria consortium developed by itself. And you can see from the picture that the reactors look green, actually very dark green. Then a couple of weeks ago, we installed the reactors in Turkey. As you can see, the system is not that different from the other one. Of course, it required, as I said, the source of the water is different, so it's secondary wastewater treatment. And these are the first pictures that we are very pleased to show, because in the very beginning, the water resulted very turbid and brown, and we were very afraid of the light penetration of, in this case, because the water was very dark. But after a few days, you can slightly see probably on the left, on the side of the reactors, some greenish that is coming out of the reactor. But in the end, this is a picture from 2 days ago, and the reactors turned very green, and we are very happy about that because they are very, very nice colors. But what we're going to do with the biomass that we will produce in these reactors? First of all, of course, we're going to evaluate the pollution level of the biomass regarding the forever chemicals and the emerging pollutants, to try to establish a mass balance, I would say, considering the pollutants present in the inlet water, in the biomass, and in the outlet. But of course, we want to perform a metagenomic analysis to establish a correlation in between which strains, species are dominant, and which is the removal rate for those emerging pollutants. Of course, these are just a slide for the procedure that we perform in the lab, and we collect the sample, we save it with some Zimo buffer. Then in the lab, the DNA extraction is performed, and thanks to PCR and sequencing with segment 16S and 18S to determine the bacteria and the eukaryote respectively, we can sequence the DNA and determine which microorganisms are present in that sample and that time. Then through some data analysis, we can draw some abundance plots, and thanks to these plots, we can correlate which microalgae or bacteria or both are responsible for the removal rates of those pollutants. Then we can move to another project, which is also takes into account microalgae, of course, and it's COSEC. It has a very long name: biogenic CO2 capture into sustainable energy carriers, a novel photosynthetic and hydrogenotrophic CO2 fixation, blah, blah, blah. It's very long. But what matters the most is that we are aiming to isolate and obtain a microalgae strain which is able to resist high concentration of nitrous oxide or phosphorus oxide, sulfur oxide present in the flue gases, with also high CO2 fixation rate, in order to have a higher production of lipid in the biomass, to use that biomass to produce either biocrude, upgrading it into liquid fuels, or biogas throughout to hydrothermal liquefaction or anaerobic digestion, so that the CO2 that has been fixed by the microalgae during their growth can be reused as fuel. So we won't have any further release of CO2 in the atmosphere. In particular, our task comprised the optimized fixation of biogenic CO2 and residual nutrients into microalgae biomass, and the strain development for improved resilience to flue gases and production of lipid-rich biomass. For these tasks, we are going to use different species of strains of microalgae that has been isolated from different sources, and they are already, since they are already isolated from wastewater, they should have already a higher resilience to higher CO2 content and flue gas content. And with these strains, we're going to perform the strain improve either the adaptive laboratory evolution to increase the content, the concentration of CO2 with which we grow the microalgae to obtain a higher resilience, or we're going to perform a strain improvement through random mutagenesis. First, we're going to determine the lethal and sublethal concentration thanks to metabolic inhibitors for the lipid synthesis. Then, through random mutagenesis with the ethyl methylsulfonate, we're going to obtain and isolate mutants which can outstand the lethal and sublethal concentration of the metabolic inhibitors. After that, we're going to isolate the strain and validate if it's actually a lipid-enriched strain through flow cytometry and Bly-Dyer method. So we have some final consideration for the microalgae. Of course, they have potential in various sectors, but the large-scale production requires improvements in economic, energetic, and environmental sustainability. We can use wastewater as a resource because it's rich in nitrogen and phosphorus, so it's suitable for microalgae growth. The biorefinery approach is very important in order to make the whole process feasible from an economical point of view. Environmental benefits are there because this approach mitigates environmental impacts from synthetic media and traditional effluent treatments. And also advancing from, if we take into account the technology readiness gap, advancing from TRL 3 to 9 is essential for a commercial deployment of these procedures and techniques. So in conclusion, basically, we can say the same. The use of microalgae with the treatment can be economically viable. More pilot and demonstration scale studies are needed to better assess the real process costs. Nitrogen microalgae consortium may be easier to manage, of course, than a specific strain, also reducing the overall cost. There are the main limitation of microalgae is the large area requirement, but probably with this green dune system, we can achieve a better ratio. There are, of course, advantages on conventional treatments because there is efficient removal of emerging pollutants and reduction of environmental impacts and treatment costs. Thank you for the attention.
Thank you very much, David and Luísa. That was great. Maybe you can stop sharing the slides and then we can have a bit of a discussion. For those of you online, please put your questions in the Q&A. It'd be great to get them. I see that we have an anonymous attendee, which didn't follow the instructions and put their name in. But the question was, Did you identify any other algae products other than biogas from your cultivation on wastewater treatment plants?
Maybe I can go on that. We just did a proximal composition of the biomass because our initial intentions were to see if the biomass had also some potential to extract lipids for biodiesel production. I didn't show those results, but the lipids content was always between 10 to 15%. And so we consider that it would not be economically feasible to move to other biofuels. We didn't check the potential for biofertilizers because on that case, it was a very highly contaminated biomass. And so we did not consider that. We only considered biofuels. Actually, most of the studies that talk about the reuse of this biomass always considers biofuels because it's like the safest way to recover some worth of this biomass because of the contamination degree. In bioremediation, we are taking a different strategy. We are going to analyze the contamination degree of the biomass. What is our idea is that there will be some degree of dilution of the contaminants in the biomass if the productivity of the system is high enough. That means that maybe if we can get a very high productive system, we can have still the contaminants there, but they may not prevent their further use as biofertilizers. With these wastes, we are not considering other uses apart from that because if you use, because we are treating like domestic wastewater, industrial wastewater, but if you use other types of wastewaters, like coming from hydroponics, for example, or from aquaculture, some other food industries, then those wastes are probably more controlled and with less toxic contaminants. And so we may be able to use them with higher value applications.
Thanks. I see there's another question, but I have a follow-on question, and it shows my ignorance here. You mentioned that you would use the outcomes of this for biogas. Does that mean you kind of end up burning it in the end to create energy? And if so, does that mean that some of these maybe heavy metals or some of these things will then go into the air? Or am I completely, I'm no chemist, so I'm asking here from a complete knowledge vacuum question.
For the production of biogas, you still need to produce the biogas. And production of biogas is done by a consortium of bacteria. So you first do an anaerobic digestion of the biomass. So you use the microalgal biomass to feed the bacteria.
Okay.
And then you have some methanogenic bacteria that those produce the biogas. So the metals will stay on the digest state. Okay, they will not be, the biogas will only contain methane, CO2, and will contain some other gases. But so the metals will not be emitted. But you are getting somewhere. It said you also produce another waste, which is the digest state.
Yeah.
But what we are doing here in COSEC is that we are using these digest states again to feed feed the microalgae. Because what the bacteria do on the production of biogas is to remobilize the nutrients that are in the form of proteins, carbohydrates, etc. They will remobilize those nutrients back into a liquid waste. And those nutrients are again back at the way they can feed the microalgae. So they are again in an inorganic way, and then they can be used to supplement the cultures. So again, you can recirculate. And the objective of COSEC is to have a zero waste process.
Okay, thanks. That was quite helpful. Okay, and then we have a real question from somebody who actually knows what you're talking about, from Gabrielle Frascaroli. saying, thank you very much for the informative discussion. We understand that one of the main challenges today is gaining acceptance for the algae sector among citizens. What do you think are the best ways to empower citizens and encourage their involvement in the academic and industrial applications of algae? Excellent question. Difficult question.
Yeah, but it's very. I don't know, Davide, if you want to jump in, please.
We should have called our friends from Work Package 7. But yes, I mean, thank you, Gabriele, for the question. I mean, it's very difficult, and actually, yes, as far as I know, all the European projects, all the projects that are being applied have this kind of work package or this kind of tasks that are demand. I mean, that, yeah, it's not important only to do research, but only to make the research viable for whomever is not from the same field. Which is the best strategy? We don't know yet. I think I don't know yet. But I mean, what I can say is that keeping also, for example, the congresses of microalgae or whatever research field that could be involved in an inclusive way for just scientists, maybe is not the wrong way, is not the correct way, because people may not be interested, but maybe they can get interested if they are allowed to jump in. Of course, in this case, I would say that who presents in the Congress or whatever have to turn down a bit, because the audience is not just made out of researchers, but could be a nice way to involve the citizen or to like to carry on this empowerment of citizens within the industrial application of microalgae, but also whatever else field, I would say.
Yeah, actually, the remediation project is doing an excellent work on this, because a big part of the remediation project is really the empowerment of citizens. So they are doing talks with municipalities, with citizens in general. Every producer of wastes is being invited to small local conferences, and they are being invited to come to the facilities at the wastewater treatment plants to visit the technology, and they are being informed of the benefits of the technology. And the same, all the partners that were chosen as the local partners have also contacts with schools. So we know there are field trips from schools going there. So they are learning what is a microalgae, what a microalgae can do, and how we can treat wastewater in a different way, and how we can benefit from it. So we need more of these projects that reach society in this way.
Yeah, I completely agree. I think actually it's so helpful. I'm not sure how many people have actually been to a wastewater treatment plant, just the usual ones. But it's really useful to go, because you really understand, you know, this is not a fun place to live or fun place to work. And then to have, so I think it would be quite helpful to actually have a field trips for, you know, for public to both of these situations, where the one is, you know, I guess there's a place for both of them, but actually I think these systems are seems very contained, and actually the moment you show some technology to people, they always get super interested, and I think that's probably a very good way of doing that. I have another question, and that's about the size of the system that these units that remediation is proposing to use. I mean, if I understand correctly, you know, you can't really put it for all of Athens, for instance. Kind of, there's a size limit to what's doable, right? And I guess that's maybe there's also then a relationship between the tertiary, secondary treatment. So if you is that correct? Am I correct in that?
Yeah. Actually, so in remediation, we are testing the two positionings of the system. So right after primary treatment and then after secondary treatments. And we are already seeing some differences. So the Turkish went green, let's say, just a week ago, but we are already looking at the aspect of the community that it's growing inside the reactors is totally different. It's going to be hugely interesting to have this. But you are totally right about the size of the installations. So we calculated, for example, for the Green Tree project, which is the one that we already have more information, we calculated that just for doing the tertiary treatment, we would need a football field size of installation, okay? And this was in Quinta do Lago in the south of Portugal, and it's a resort, and it's, let's say, one of the most expensive grounds in Europe. So they are not going to take out a golf course to do wastewater treatment with microalgae. Forget about it. But one of the new things of this new legislation is that the size of the populations that can continue without wastewater treatment has decreased from 2,000 population equivalents to 1,000. That means that a lot of countries in Europe that are not doing a wastewater treatment legally, let's say, so legislation allows them not to do because they were very small populations. Now they have to think about something, installing some sort of wastewater treatments. Usually those small populations are in rural areas where space is highly abundant. So I think that the easiest way to put this technology out there is within these populations. Microalgae this system, and especially using these natural developed consortia, do not require a lot of manpower and also a lot of specialized knowledge. You need some knowledge or some specialists to keep an eye on things, but the day-to-day does not require that much special knowledge. So this would be really a good alternative for these remote locations that are not having wastewater treatment. And because then also the degree of contamination, because they don't have a lot of industrialized wastes, the biofertilizers would be a good way. So you do really a circular economy on this place.
And actually, maybe this is also a good time to point out that actually under remediation, they are now looking for other sites to institute these systems.
Associated regions.
There we go. In associated regions, they can actually get some money to help set up these projects in other sites. So if you're interested, then you really should try and contact the people. You can contact us or contact the speakers. And I see Gabriela has just posted one more, and then I think that's it. So she says, I agree with you, on the need to bring rocket science, which is not closer to the people. A more specific question regarding the use of consortia. Do you think it would be possible to cultivate separate species, separately specific robust species close to the reactors by naturally harvested and specifically selected? For example, through the recirculation of biomass with the addition of specific strains, we could partially inoculate the system when its performance declines or when it collapses, or when its collapse is imminent. I don't know if you saw that question. It was quite a lot.
Yeah. I'm reading it. We could consider something like that. Actually, the system is quite fast in the recovery, and let's be frank. Even bacterial wastewater treatments collapses from time to time. Okay, so when you have huge climate variations, for example, an episode of heavy rain, you can have some difficulties, and the system is not working correctly. Yes, it would be a nice idea to have like a backup, a back plan. yes, and this could be. In terms of choosing the strains, that is a bit, it requires more control of the system. And we are using that approach not with domestic wastewater, but with COSEC when we are using other wastes. There, yes, we are going to control the strains that we use. But the idea also is to choose the strains and improved strains because we want to promote the reuse of the biomass in a different way, so in a more efficient way. And then we need to select the strains. For domestic or urban wastewater treatment, I don't find that as feasible.
Yeah. All right. Thank you very much. I think we've just run out of time. So I'm hoping you can see the slide that I have with our next webinar. Is that correct? Yes. Yes. So I just want to thank you both. It's been really interesting. I learned a lot, as I usually do during these webinars. And I really look forward to the outcomes of the remediation project and all of the other projects that you mentioned. And now I think it's only for me to say that our next webinar will be on the 15th of May, and it'll be on the deep pelagic biology, biodiversity, and carbon flux, which is linked to our very new document, the Future Science Brief number 12 on the deep sea research and management needs. And the webinar will be by Henk-Jan Hoving from Geomar. So with that, I just want to thank again our speakers, and yes, thank you very much for your time, for bringing your work to our attention, and yes, looking forward to the next one. Thanks.
Thank you very much for listening, and thank you very much for
thank you. Thank you.
Bye bye.